Today we're going to be discussing developmental networks and how regulation of gene expression occurs. Let's discuss this with an example, the thorny dragon, which produces spikes over parts of its body. The developmental network for the spike expression looks like this. An arrow means that the action is enhanced, while a block means that the action is repressed. I like to think of it as a really complicated friend group where different friends encourage certain behaviors, while others oppose those behaviors. Now, this looks a little complicated, so let's see how this gene sequence and some mutations of this sequence affect the phenotype of our thorny dragon. Let's take a look at our wild-type strain. In our wild-type strain, S promotes P, and P will in turn block I. If I is blocked, that means that K can promote E, and E will produce the spikes. Therefore, our thorny lizard will have spikes. Let's take a look at a strain where the P is missing. In this strain, the P is missing, which means that I will block K, which will in turn not be able to promote E, which means that our spikes will not be produced. Let's take a look at a strain where the I is deleted. If the I is deleted, that means that there's nothing stopping K from promoting E, which means that E will in turn make their spikes, leading to spikes on our thorny dragon. Now let's take a look where we have two different mutations, one where the I is missing and one where the E is missing. For an example like this, we will be looking at simply the second mutation, or the one that's closer to our end product. In this one, the E is missing, and if we can't have our E, that means that we simply cannot produce any spikes at all. Therefore, our thorny lizard will have no spikes. And finally, let's take a look at one more example. In this example, the S is missing and the K is also missing. Again, we're going to be looking at the second one closer to our end product. If K is not there, that means that K cannot promote E, which means that we will simply have no spikes. Now, we've discussed how different mutations in our gene sequences could lead to different phenotypes, but we can actually also have different phenotypes from the same genome sequences. Let's take a look at an example. Histone is when an acetyl group is added to the lysine residue in a histone tail, leading to a neutral charge. This neutral charge loosens the interaction between the histones and the DNA because there is less attraction between the negative charge of the DNA and the now neutral charge of our histone. The result of this change in charge is actually increased transcription, as it is easier to access the loosened DNA. In our thorny dragon example, if histone acetylation occurred in some cells, there might be higher transcription of genes that work to repress the spikes on the skin, which would mean no spikes for those cells. Other cells in the thorny dragon's body might not undergo histone acetylation, meaning that no spike repressor proteins would be transcribed, leading to the formation of spikes on the skin.